Hey guys! Today we're going to continue with topic 2.8, cellular respiration. And I just want to remind you the last lecture that we had talked about anaerobic respiration. So that is the breakdown of glucose without oxygen. And we talked about the different ways that that can happen. We talked about the first step, which is glycolysis. And then we talked about the second step, which could be a couple of different things. It could be lactic acid fermentation, like what happens in your muscles when you work out, or it could be alcoholic fermentation, right? And in both those cases, you don't actually get more ATP, but you reset the cell for glycolysis to happen again. So now we're going to move on and we're going to talk about aerobic respiration. So we're going to start with understanding number 4. Aerobic cell respiration requires oxygen and gives a large amount of ATP from glucose. And there are 3 steps that are involved. There's glycolysis again, there's the Krebs cycle, also known as the citric acid cycle, and there's step 3, which is the electron transport chain. So I'm going to go through this step by step and show you all the nitty-gritty details. But I don't want that to scare you. I do that because I want you to kind of be able to visualize the process. The important thing is that you understand what the reactants are and what the products are for each step. And if you can do that, then you'll be fine. Something else I want you to notice is that quite often, the products of one step become the reactants in the next step. And that's important. When we talk about metabolism, we talk about chemical reactions in living beings happening in a predictable and organized way. And just like when you're baking a cake, you have to follow the recipe in the right order, right? So these chemical reactions have to happen in a certain order because the products of one step become the reactants in the next step. And you can see that here too. So we'll take it step by step. Remember that glycolysis is the first thing that happens, whether it's anaerobic or aerobic, and that happens in the cytoplasm. The next step is going to be the Krebs cycle, and the third step is going to be the electron transport chain. And we'll talk about what these reactants and products are as we go. We basically have 3 different classes of molecules. We have energy carriers, we have waste products, and we have carbon compounds. So let's take it step by step. Okay, first things first, just like last time, glycolysis. Here we have our glucose molecule, right? 6-carbon ring. And this is occurring in the cytoplasm of eukaryotes. What happens? That glucose is going to be broken down using 2 ATP, some NADH, which is an energy carrier molecule, and some enzymes represented by our scissors. So in the first step, those ATP molecules are going to be used with some enzymes to break that carbon molecule. Now that phosphate group adds on to the end of that carbon molecule because remember, we want to make sure our carbon molecule is stable. Each carbon atom has to bond 4 times to be stable, right? So to make this stable, we have to add that phosphate group. Same thing at the top. And that's how those ATP molecules become ADP. What we end up with are 2 3-carbon molecules called pGal. Now you're not going to need to know pGal, but just so that you understand that there are sub-processes and sub-steps. We have some enzymes that are going to add more phosphates onto the end of those molecules to make them stable. And now we have some NAD plus that's going to come in, and it's going to steal some of those hydrogen because remember, these aren't just carbon molecules. We started with C6H12O6, right? You can see the carbons, but you can't see the hydrogens. They're hiding in there. So these NAD molecules are going to come steal those carbons or steal those hydrogens, kind of like stealing cookies from the cookie jar. And they're going to take those to the mitochondria. Now we're going to pull these phosphates back off. And when we do that, we're going to convert 4 ATP and we're going to charge them back into ATP. So, what we have left over are 2 molecules of pyruvate. These you do need to know. So as a result, we have a net gain of 2 ATP. I say net gain because remember, we had to spend 2 to get 4. So we started with 1 molecule of glucose, 2 ATP, and we ended up with 4 ATP, 2 molecules of pyruvate, and 2 NADH. So now what? Well, that pyruvate is really important. 2 different things can happen. Those pyruvates can get broken down further. That's the aerobic pathway that we're going to learn about today. Or fermentation happens, right? And we talked about this pathway last time. Now we're going to talk about this pathway. Okay? All right. So let's start with the Krebs cycle, otherwise known as the citric acid cycle. So now that these pyruvate are hanging out, right? They're also going to be taken to the mitochondria. So let's zoom into the mitochondria. Here we have our pyruvate, and we're inside the cristae or the matrix inside the I almost said chloroplast, the mitochondria. All right, we're in the mitochondrial matrix. That pyruvate is going to be broken down into acetic acid. We have NAD plus that's hanging out inside the mitochondria, and it's going to remove more hydrogen. It's going to steal more cookies from the cookie jar to create NADH. Now, the whole point of NADH is to carry hydrogens away. When that happens, we release a molecule of carbon dioxide, see, CO2. So when you are breathing out, the carbon dioxide that you are breathing out is a result of this process. Carbon dioxide is a waste product as a result of the Krebs cycle in cellular respiration. That's a good test question. The next step, coenzyme A is going to bond to acetic acid. There's our coenzyme A. It's going to bond to our acetic acid, and that creates a compound called acetyl-coenzyme A. Next, citric acid is going to form when acetyl-coenzyme A bonds with a 4-carbon molecule from the last Krebs cycle. So here we have a leftover carbon molecule from the last cycle. See how it's like blinking? And that is citric acid, C6H8O7. Next, this molecule is going to be broken down into a 5-carbon molecule. Now, you're probably wondering, why do we keep breaking and reforming this, right? Because that's all this process is. We just seem to be like taking a carbon compound and breaking it and reforming it and breaking it and reforming it. Because remember, a chemical bond is nothing but stored energy. So Cellular respiration is the process of rearranging the molecule so that we can break it to get more energy out of it. Every time you break a bond, you're releasing energy. So this entire process is rearranging the molecule and breaking it, and rearranging it and breaking it to keep getting as much energy out of it as you can. We have NAD that's going to remove hydrogen to make NADH. We call these energy carrier molecules because they're going to carry energy in the form of these hydrogen ions. And we release some more CO2. Next, this 5-carbon molecule is going to be broken into a 4-carbon molecule. And we're going to steal some more cookie jars, or cookies from the cookie jar. We're also going to make a little bit of ATP. So here's some ADP that's waiting to be recharged. It's hanging out in the mitochondria, waiting to be recharged. We're going to use this carbon molecule as a template to help us fuse that. And we're going to release some more carbon dioxide. So next, we rearrange that 4-carbon molecule. We have NADH, which is going to come steal another hydrogen. And we also have FAD, which is like, it's fatter than NAD. It's going to steal 2 hydrogen instead, right? So here's our NAD+. And here's our FAD. It's going to steal 2. And for those of you that had me freshman year, this probably sounds familiar to you because like this is literally the exact same PowerPoint that we used your freshman year. So you learned all this stuff already. So don't be afraid of it. You got it then, you'll get it now too. Next, our acetyl coenzyme A is going to bond to that 4-carbon molecule and recreate citric acid. Remember, this is a cycle. And there is our molecule that we're going to use in the next cycle. Here's our coenzyme A. We're going to form citric acid, and then the process repeats. Right? And don't forget, we have that other pyruvate molecule over here too. So we started with 2 NADH plus that came from glycolysis. We started with some ADP that needed to be recharged. We started with pyruvate that came to the mitochondria. And as a result, we got 8 NADH, 2 FADH2, 6 molecules of carbon dioxide, and 2 ATP. So just like last time, the products of this reaction, all these things, except for the CO2 because that's waste, right? But this NADH and this FADH2, we're going to use this in the next step. These become ingredients for the next reaction. That reaction is the electron transport chain, and that occurs in the membrane of this cristae, this fold right here, right? We used to call the mitochondria the maize bean. I think Owen started that, right? The maize bean. So now we're looking at the edge of the maize in the maize bean. Okay. So those NADH that we created during the first 2 steps and the FADH that we created in the last step, they're going to deliver those hydrogen ions and electrons. Now you notice when these molecules release those hydrogens, it's also releasing some electrons. These electrons are going to activate protein channels, right? This is active transport. We're using energy to activate these channel proteins through the membrane, and they're going to pump these hydrogen ions out. Here we have ATP synthase. Now you know it's an enzyme because it ends in ASE, right? All enzymes end in ASE, and enzymes are used to build things up and break things down. Well, this enzyme, its name is ATP synthase, like to synthesize. So you can probably guess that this protein, this membrane protein, is also an enzyme that creates ATP. These hydrogen ions, right? Now we have a concentration gradient where we have a high concentration out here and we have a low concentration in here. So these hydrogen ions are going to diffuse through ATP synthase, right? And that's going to power this enzyme to create ATP by adding this phosphate to this one. It's kind of like a windmill or like a water wheel. And this process creates up to 34 ATP. Now, that's a lot. Remember, glycolysis only gave you a net gain of 2 ATP. We didn't make very much ATP during the Krebs cycle either. We made like 2, right? So that's not a whole lot. It is this molecule right here, ATP synthase. Without this molecule, multicellular life on our planet would not exist because it has the capacity and the ability to create a ton of ATP. So be thankful for your maize bean because without it, we would still be prokaryotes, single-celled organisms hanging around one at a time. With all this extra energy, that allows organisms to start spending that energy and differentiating and doing more things. Instead of just being one cell, now you can be an organism that can afford more cells that do different things, right? So the evolution of multicellular life really depends on this molecule right here. It also goes back to endosymbiotic theory that we talked about, because remember, these mitochondria, they used to be their own organisms. They used to be their own prokaryotes. Some prokaryotes sucked up a mitochondria, and instead of digesting it, it was like, yo, hey, you are pretty good at this ATP thing. So why don't you just live inside me, right? I will make sure that you get the stuff that you need, and you can create butt tons of ATP. And it is after that point that you start to see eukaryotes in multicellular life. It's pretty amazing that it's all down to this, right? Okay, I digress. Let's get back. Now, we also create water waste during this process. Remember, this is aerobic respiration, so it's in the presence of oxygen, right? Those hydrogen ions that have diffused back in, they combine with some of the electrons that have been used across the electron transport chain and oxygen molecules to create water. So when you breathe out, there's water vapor in that breath. That water is the result of water that's produced during cellular respiration. So the carbon dioxide and the water that you breathe out when you exhale, they come from different parts of aerobic respiration. The carbon dioxide comes from the Krebs cycle. The water comes from the electron transport chain. So let's review. I like this diagram because it kind of simplifies everything. I realized that those steps were super complicated and there was a bunch of big compounds in there that you probably aren't familiar with and you're like freaked out about knowing about them. But I don't think you should focus on that. I think if you can complete this diagram and explain it properly, then I think you need everything, or you have everything you need to do well on a skills question about respiration. Okay? So let's fill this in together. We have glycolysis, which is the first step, right? And that's where we turn glucose into pyruvate. And this happens in the cytoplasm of a cell. Now, one of the products of glycolysis are NADH molecules. and their job is to carry NADH, well, they're NAD plus, and they carry that H in those electrons. Their job is to carry them for later. We also produced 2 ATP. And we call this, I think it's in here. I don't want to write it if it's in here. I think we call it, well, I know what we call it, but I'll just leave it. Yep, there it is. Substrate level phosphorylation. And all that is a big fancy word that says we create ATP by adding a phosphate, phosphorylation, right? This word, phosphorylation, it means to add a phosphate. Substrate level phosphorylation. It means we're taking ADP and turning it into ATP. All right, so if you want to write that, you can write that. Okay, so we started out with 1 molecule of glucose that we got from our food that we ate. We turned it into pyruvate. In that process, we made some NADH, which we're going to save and use for later. And we created some ATP. Now remember, it's a net of 2 ATP because we had to spend 2 to get 4. This is the same process that happens in both aerobic and anaerobic respiration. It's after this step that things get different. In aerobic respiration, we're going to move on to the citric acid cycle, which occurs in mitochondria. So during the citric acid cycle, we take that pyruvate and we convert it and change it and shuffle it around and break it and shuffle it around and break it. And the result is we get energy carrier molecules, NADH and FADH2, that we're saving for later that we're going to use. We also create ATP. I think we create 2, right? And we call that substrate level phosphorylation. Now, we also create some waste products that we need to add. The waste product of the citric acid cycle is CO2. Now, for our third step. Oxidative phosphorylation and the electron transport chain, sometimes called chemiosmosis. Don't freak out about the big words. It's okay. Oxidative means in the presence of oxygen, right? You guys took chemistry last year. You should know what oxidation is. Phosphorylation, same thing, turning ADP into ATP. And we have the electron transport chain. So we're going to use the products of our other reactions. We're going to use these electron carriers and these NADH molecules. We're going to use these FAD, and we're going to use that to produce 34 ATP. We're also going to create a waste product, and that waste product is water. So, in review, aerobic respiration, which is considered these 2 steps, right, creates a total of 36 ATP. That's a lot more than anaerobic respiration, which just gives you 2. So again, we owe the existence of multicellular life to this little guy right here, the maize bean, and in particular, the ATP synthase in the third step in the electron transport chain. So, pretty important. Go maize bean. Oh, we call that oxidative phosphorylation. Yeah. Okay. So at this point, you should be able to do a practice quiz, right? You should be able to tell me how much ATP is created during glycolysis, how much ATP is created during the Krebs cycle, and how much ATP is created during the electron transport chain. And you should be able to tell me where the 3 stages of cell respiration occur. You should also be able to put these different processes in order. And it might look scary, but it's not. If you follow a certain strategy, it makes it a lot easier. Instead of trying to put them in order from front to back, if you can first just put them in the proper stage. So is it glycolysis? Is it the Krebs cycle? Is it the citric acid cycle, which is the same thing as Krebs? Is it electron transport chain? If you can just get it into those first, then it becomes much easier to put them in order. So like ATP produced through ATP synthase. Well, that happens during the electron transport chain. Okay. ATP and pyruvate are created. All right, well, pyruvate is created pretty early on, right? And that's glycolysis. Okay, hydrogen ions are pumped out of the matrix. So not NADH, just the hydrogen ions themselves. That's the electron transport chain. Oh, acetic acid bonds with coenzyme A. Well, it's acetic acid. That sounds like the citric acid cycle to me. So we'll just put Krebs. Okay. Citric acid is created. That's also Krebs. Okay. Glucose is broken into pGal. Glucose. Okay. Glucose is like our very first reactant, right? That's the very first thing. So that's got to be glycolysis because that's like our starting point. Did I spell that right? Glycolysis. Okay. And then ATP, NADH, and FADH2 are created. Okay. Well, ATP is created like in each step. NADH is created in a bunch of different steps, right? Both glycolysis and the citric acid cycle. FADH2, though, that's the key. FADH2, it's used in ETC, right? It's used in the third step, so it's got to come before that. So that means it's got to be, pretty sure that's Krebs. Okay. Now that you know what each step, what stage it's in, now it's much easier to put those in order, and I'll let you do that yourself. Okay? One more thing before we go. I want to summarize this entire process for you so it becomes a little more clear. When we're talking about cellular respiration, we're really talking about 4 different types of concepts. We have organic molecules, right? That's any molecule that contains carbon that's produced by a living thing. We have processes that happen, right, when we're trying to get energy out of those molecules. We have energy molecules that their entire job is to hold on to energy and store it. Because remember, the process of cellular respiration is converting the energy that's stored in glucose into a form of energy that your cells can actually use. Your cells can't spend glucose, they can spend ATP. So, in order to make ATP, which is an energy molecule, right, we have to break organic molecules to access that energy that's stored in those chemical bonds, because all chemical bonds are is stored energy. And of course, we're going to create some waste in this process. Waste is always a thing. So if we look at an overview of the entire process and keep an eye towards these 4 different types of concepts, we have organic molecules, we have processes that happen to these molecules, we have energy carrier molecules, and we have waste. We can kind of summarize and make a really simplified diagram that helps order things. So we're going to start with our organic compound, and that's glucose. C6H12O6. We're going to begin to break that molecule down using the process of glycolysis. That's a process. It's the first step. Now, the next step depends on whether you have oxygen or not. We have 2 products from glycolysis. We have pyruvate, which is another organic molecule, and we have 2 ATP, which is an energy molecule. What happens now depends on whether you have a mitochondria and whether you have oxygen. If you do have mitochondria and you have oxygen, you have aerobic respiration, which gives you a net of 36 ATP, and it gives you waste products of carbon dioxide and water. Right? And the steps are Krebs and etc. If you don't have oxygen and you don't have a mitochondria, then you're going to go through the process of fermentation. If it's lactic acid fermentation, you're going to get waste products of lactic acid. If it's alcoholic fermentation, you're going to get waste products of ethanol and CO2. You're also going to recreate NAD so that the cell can start glycolysis over again and go through this process again. I hope that summarizes it for you. So skill number 1 is the analysis of results from experiments involving respiration rates in germinating seeds using a respirometer. Now, we're going to do a different kind of experiment. This one's kind of boring. You take a glass tube and you soak some peas in water, and you have other peas that are not soaked in water. And what we're going to measure is the amount of gas that's produced. You have some cotton underneath that's not going to absorb the water. And then you have some cotton that's soaked with potassium hydroxide at the bottom. You also have a control, which has the same setup, but there's beads instead of peas, right? And the reason we use peas is because they will start respiring on their own once they are reconstituted. So these guys are dormant, right? They're not going to be respiring. These guys are soaked, meaning they have water, and they are going to start respiring. This is a rubber stopper, and this is a tube that allows gas to travel. So this is just one setup. There's a lot of different setups. Things that you should be aware of. The potassium hydroxide at the bottom is going to absorb carbon dioxide that's produced. So we put this whole thing in water, and as the seeds start to use oxygen and that carbon dioxide is absorbed, the gas levels in this tube decrease. So water is pulled into the pipettes, and that's how we can measure. So you can think of it as like water being pulled in because the gas is disappearing. And it gives you a rate of respiration. And what you should see is that the germinating peas, the peas that are actively growing and going through mitosis, they're going to be using cell respiration, right, to power that process, to produce energy so that they can go through mitosis. And the dry peas aren't, so you shouldn't see any change. And then this is just your control. It's kind of an awkward setup. I don't particularly like this particular setup. I think it's complicated and kind of unnecessary. But it's one that you have to know. You have to understand this process. But Zane and I will come up with a different lab. People at home, I'll have a virtual lab set up for you next week. And I think Zane is going to try and do something in person. All right. So at this point, you should be able to answer these questions. I'll put up a quiz or review quiz for you to test your knowledge. And have a good weekend. Talk to you soon.